guys, my name is Nako Nakatsuka. I'm a fourth year chemistry PhD student at UCLA, and today I'll be helping you guys out by going over some general chemistry concepts. And good luck with the course. So now I wanted to introduce you guys to the 20 common amino acids, and you will probably have to memorize all of these different structures, all 20 of them, so make sure you start doing that. And so let me read this to you really quickly. Amino acids are the building blocks of proteins in living organisms. There are over 500 amino acids found in nature. However, the human genetic code only directly encodes 20. Essential amino acids must be obtained from the diet, whilst non-essential amino acids can be synthesized in the body. So you can tell the difference between the two using this chart key. So non-essential ones, which can be synthesized in the body, are these solid circles. On the other hand, if they're essential, where you have to consume it, is this dotted line. So for example, isoleucine, leucine, valine, phenylalanine, tryptophan, all of these have to be gained from eating. And so let's see. We also have aliphatic versus aromatic, acidic versus basic, and also hydrolyxic, and sulfur-containing and amidic, where you have an amine group. And so I think this is a really nice way to separate all of these for you, so you can kind of group them together based upon their functional groups, based upon their properties. And so two main things you should split up the amino acids is by hydrophobicity and also acid versus base. And so as you can see, if we go through each of these structures, they only differ by one thing. So for example, in the case of alanine, it has a methyl group right here as the R group. In the case of glycine, they don't draw it on here for you, but it's a hydrogen for the R group. In the case of isoleucine, it's this part. So as you can see, for each of the amino acids, it has the central backbone that I showed you before, and then it just has the extra R group. And so just make sure you familiarize yourself with the different families, how each one has these different three-letter abbreviation, which is often used when characterizing peptides, which I will show you in a little bit. So as I mentioned before, amino acids can form peptides, and they do so via peptide bonds. And so this is a brief overview of how they're formed. And so we have one amino acid right here, and another amino acid right here with their different R groups. And then we undergo a dehydration reaction where this OH group from the carboxylic acid on the C terminus of this amino acid reacts with this hydrogen on the N terminus of this amino acid. Dehydration means you get rid of it as water, and you end up creating a bond between this carbonyl group and this NH group. And thus this C double bond O and H is the peptide group. And so if you look at this example, this is glycine, serine, and valine, which are three amino acids side by side. This is called a tripeptide because there's three of them. And you can see that each of these peptide bonds are indicated in red. And so you have the C double bond O and then the NH. And this is going to be the repeating unit for every bond you make between amino acids. The only thing that's different for all of these is the R group. So in this case, for serine, you have this R group. For valine, you have this R group. And then for glycine, you have this hydrogen that's not indicated. So peptide bonds can actually influence structure. And so I can draw you a quick diagram right here comparing a trans and cis isomer of the peptide bond. And so in this case, if I have two things attached like so, then the amino acid chain is on opposite sides of the carbon-nitrogen bond, right? This is going up, this is going down, and thus this is trans. On the other hand, what if we had cis? In this case, the hydrogen would be here, the carbonyl would be here, and now the amino acid chain is on the same side of the carbon-nitrogen bond, and thus this is cis. So the torsional strain is less for trans in comparison to cis. So the equilibrium favors the trans isomer by approximately 2 kilocals per mole. So ultimately, the amide is conjugated, which is something we talked about before when we went over conjugation, right? And so if I drew it out like this, we have the oxygen right here with the 2 lone pairs, and the double bond could be on either place. And so this oxygen would have a slightly negative charge, and this nitrogen would have a slightly positive charge. And so the amide is conjugated, and the conjugation results in a barrier to rotation around the carbon-nitrogen bond. And thus, all of these peptide bonds are planar.